Hi everyone, in this video we're going to go over aerobic respiration, one of the components to metabolism. In the color black are the molecules that are going to go into any given process, red is the process, and green is what we get out of those processes. So at the top we have our glucose molecule, which we're going to put through glycolysis, and from that we get 2 NADH and 2 ATPs. After glycolysis our glucose has become pyruvate. Pyruvate is a 3-carbon molecule, and we have 2 of those because glucose started out as a 6-carbon molecule. So basically we cut it in half to make our pyruvate. Afterwards we'll take that pyruvate and put it through pyruvate decarboxylation to get acetyl-CoA. And in doing so we create 2 NADH molecules. The acetyl-CoA will go through a process known as TCA, Krebs, or citric acid cycle. They're all the same thing. From that, we get 2 ATPs, 2 FADH2s, and 6 NADH molecules. Now, you'll hear Dr. Bazan often say that after aerobic respiration, we get 32 ATPs. But from this, you can see that we only have 4 ATPs. So how do we do this? We collect the NADH molecules and the FADH2 molecules, and we put them through something called the electron transport chain. In the electron transport chain, for every NADH, we get 2.5 ATPs, while for every FADH2 molecule, we get 1.5 ATPs. When we add up the total amount, we get 25 ATPs from NADH, 3 from FADH2, and then the 4 that we had through the other processes over here. And that's how we get our 32 ATP. Now on your guys' test, Dr. Bazan will say something like, how many ATPs do we get from aerobic respiration? He'll throw out numbers like 32, 27, 6, and 5. It's going to be the number that's in the 30s. He may or may not choose 32. He might say 36, 34. So what's important to remember from this is that you're going to make somewhere in the realm of, depending on what book you read, 28 to 36 ATPs. But just know that he's not going to say 32 and 36. Not going to happen. It's going to be really clear cut and dry what the answers are. The numbers aren't even going to be close. So let's look at how the electron transport chain transfers energy from NADH and FADH2. As we take a closer look at the electron transport chain, we can see that there's a lot going on here. This illustration mostly just helps me to depict the point that NADH and FADH2 are generating a lot of ATP. And the only electron carrier that you need to worry about is cytochrome C, which Dr. Bazan will cover in lecture, but you guys will also look for in your biochemical lab when you do the oxidase test. At the bottom we can see that we have our 2 high energy yielding molecules. NADH, which generates 2.5 ATP for every 1 molecule, and for every 1 molecule of FADH2 we get 1.5 ATP. So how does this happen? It happens by placing NADH and FADH2 through electron carriers, which will oxidize them to NAD+ and FAD+ respectively. As we displace these electrons, we also displace protons from one side to the other. And as these electrons are passed down the chain, more and more protons are also pumped from the matrix to the intermembrane space of the mitochondria. Now this does also happen for prokaryotes, but it happens within their cell wall. So as you can see here, because this happens in mitochondria, this is dealing with eukaryotes. Again, the concepts are the same, but just know that the location where this occurs is different for prokaryotes and eukaryotes. Now, we also have our final electron acceptor here, which is oxygen, which kind of acts as a magnet for the electrons. It is a final electron acceptor, and when mixed with some protons, it will eventually make water. This is because not all the protons are pumped to the other side. There are protons on this side that will react with the oxygen in order to make water. Now, we have a bunch of protons on this side. And how do we get rid of those? We get rid of them through chemiosmosis. which is a process in which chemicals move down their electrochemical gradient in order to generate ATP. And that's what happens here. The protons will flow through the ATPase, which is this structure here in blue. And what this does is it allows for the protons to pass through it. And as the protons pass through the ATPase, it joins ADP, an inorganic phosphate, in order to generate ATP. So as you can see, NADH displaces more protons because it starts higher up in the chain, while FADH2 starts farther down the chain. So it would only make sense that NADH, since it displaces more protons and more protons pass through here, more NADP is brought together with inorganic phosphate to make ATP. And this happens for every NADH and FADH2 molecule. So going back to the original equation, we produce 10 NADH molecules from glycolysis all the way to Krebs. And we've produced 2 FADH2 molecules from Krebs. So we have to do this for those 10 NADH molecules, for those 2 FADH2 molecules. So that's how we generate so much energy, is that we've pumped so much protons to one side that they have to flow down their electrochemical concentration gradient. And as they do that, they're generating all this ATP. These are why these molecules are high energy yielding, because they cause this ATPase to rotate, which causes the ADP, an inorganic phosphate, to come together in order to make the ATP. And I understand that there's a lot going on in this picture. If you guys have any questions, please feel free to shoot me an email, and I will definitely get back to you guys about this. And with that, you guys have your aerobic respiration. So just to summarize everything, we have our glucose that we put through glycolysis in order to give us pyruvate. In that process, we created 2 NADH molecules and 2 ATP molecules. That pyruvate then goes through pyruvate decarboxylation in order to give us acetyl-CoA. That process yields 2 NADH molecules. The acetyl-CoA will then enter the Krebs cycle, which yields 2 ATPs, 2 FADH2s, and 6 NADHs. The NADH molecules and the FADH2 molecules will then go through the electron transport chain, which converts those high energy yielding molecules into ATP. Now, if you guys have any questions about this, please feel free to shoot me an email, and I look forward to hearing from you guys.